All right, Dr. McNeil, Alex Shaw behind the camera. Eukaryotic gene structure transcription. So we just got done talking about prokaryotes, bacteria, simple stuff. Eukaryotes are substantially more complex. So in eukaryotes, there are multiple RNA polymerases. That's why I wrote mRNA poles. Sorry, RNA poles. Not all of them write mRNA. And we have three major RNA polymerases that have been well studied, especially in things like yeast. And each one of those RNA polymerases does a different major function. RNA pol 1, we're not going to worry about very much. Mostly transcribes our RNA genes. These are genes that code for ribosomal RNA. Because the ribosomes are hugely composed of RNA. It's a ribonucleic protein complex, so it's got a lot of RNA in it. And we have a special RNA pol that mainly transcribes that. Okay? We have RNA polymerase 2, RNA pol 2. That guy mainly transcribes mRNAs. So they make RNA for protein coding genes, mainly. And we have RNA polymerase 3, which mainly helps do transcription of transfer RNAs and lots of small RNAs that, if we go fast enough through the semester, we'll be able to talk about at the end. So, what's the difference between bacterial RNA polymerases and our RNA polymerases? Well, there's a lot of differences. Here's a couple. One is that eukaryotic polymerases are more processive than prokaryotic. What does processive mean? Processive I'll put processiveness is how many base pairs it's technically not a pair. How many bases an enzyme can write before it falls off the DNA. So, eukaryotic RNA polymerases are much more processive than prokaryotic. Why? Well, we can do about 10 to the 6 bases or more. Prokaryotes can only do about 10 to the 4. So we can do millions of bases. They can only do thousands. Why is that? Well, the largest eukaryotic gene there's two of them that are about this size, are 2.3 million bases, megabases, that's million bases, are 2.3 million bases long. The entire E. coli genome is only 4.5 million bases. So we are hugely more complex. Our RNA polymerases have to be able to stay on the DNA for a lot longer. Our polymerases, partially because of their bulkiness, partially because of the complexity of our genome, move much slower than bacterial RNA polymerases. So ours do in the neighborhood of about 40 nucleotides per second, as you see. Whereas prokaryotic do about 70 nucleotides per second. So prokaryotic RNA polymerases are a lot faster. Okay? So, there's some major differences. The last major difference that's probably really worth considering is that eukaryotes, like us, do not use sigma factors. Okay? And we use a lot more greater than 20 proteins for the RNA polymerase hollow enzyme. If you recall, bacteria only use about 6. So we are far more complex.